Okay. So last week we already started talking about, or not last week, but Monday, we already started talking about different ways that reactions happen inside our body, how enzymes are helping out to make sure that those metabolic reactions are able to occur, sometimes simply by reducing the activation energy, but also by those linkages of anabolic and catabolic processes so that even though some of the reactions may not be energetically favorable, they're still able to happen, they're still able to occur by linking them to highly energetically favorable reactions with a delta G that's really negative. So as long as the sum of all the reactions in a metabolic process is negative, you are going to be okay, and you're still going to have an energetically favorable outcome. So what do we utilize? Well, both plants and animals, at the end of the day, we are using sugars as our main source for fuel. Plants obviously make this by themselves using photosynthesis, using those inorganic materials, sunlight, all that good stuff. Animals, on the other hand, will obtain these through the foods that we eat. And then they have to break it down for its components so that they can get those high energy chemical bonds to release the energy that is then utilized in the metabolic processes. So regardless of whether an organism makes its own food in the form of, plants making sugars from carbon dioxide by photosynthesis, or whether they are in consuming it, they still have to, at the end of the day, oxidize it, break it down, and then utilize that energy for their own consumption. So this process obviously is going to happen in both plants and animals. And to do this process, it is, again, not necessarily going to happen just out of nowhere. Enzymes have to be utilized in a very long series of controlled reactions in order to get stored energy from those bonds that are in that sugar molecule, right? So cells use the enzymes and they break it down, not just by breaking it down once, but breaking it down into smaller molecules, and each time smaller activated molecules through a series of reactions, which allows each time to have some free energy released, and that energy can then be stored in the form of activated carriers, whether it's ATP, NADPH, NADH, whatever that may be, and you can utilize it a lot better. So it's not just a simple, let's just destroy the sugar molecule. It is, and release all the energy all at once. It's a very controlled process. It's a very stepwise process, and that allows us to utilize the energy a lot better, a lot more efficiently. It's going to take it all in little tiny packets and be able to utilize it a lot more. All those little activation energies that are going to be utilized in each one of those reactions are going to produce heat for our body, temperatures, right? That's going to provide our body with its metabolic rate as well. And all those little packets of energy that are released at each point as the bonds are broken down are stored in activated carriers to be utilized downstream. So when foods are broken down, there are three different stages. They are, it is a catabolic process. And this is basically the process of taking that big, long macromolecule chain, whether it is a protein, polysaccharide, or fat, and taking it, digesting it, breaking it down into its individual monomer form. And these stages of food catabolism are called digestion, which is the first step where you're enzymatically breaking down the food in intestines and taking all the energy with the help of lysosomes inside the cells. Then you have glycolysis, where you are taking the glucose molecules that are present within your food and breaking them down. So glucose is a 6-carbon sugar. In that first part, you're going to take that glucose and you're going to break it down into 2 pyruvate molecules, so 3-carbon sugars that are no longer cyclical. And then the third part is the citric acid cycle and the electron transport change, which is where the bulk of the energy is going to be released from your system, okay, from those sugar molecules. So that first part of digestion, which is the enzymatic breakdown, it's going to take both all of these things, whether it's proteins, polysaccharides, fats, and it's going to break them down in a way that they can be put into this cycle at some point. Not everything is going to go straight through all of the steps, right? So sugars, obviously, they are going to go to glycolysis and they are going to go to pyruvate and acetyl-CoA. Amino acids, on the other hand, they are going to have, the protein chains are going to get broken down so that you actually come into an intermediate step. They are not going through glycolysis. They are going to be breaking down into simpler subunits with acetyl-CoA, and then they are going to be coming in either at the citric acid stage, acetyl-CoA, or pyruvate. So that's where they are coming into the equation. Similarly, the fats get broken down, right, with, again, coming into the acetyl-CoA cycle and not at the glycolysis step. And then past that point, no matter what molecule you had up front, it's the same. It's going to still be producing the same types of energies. And it's going to be releasing all of that through the citric acid cycle and electron transport chain. Okay. So glycolysis, which is the very, very first part of glucose metabolism, essentially means splitting of sugar. That's the only part of the cycle that happens in the cytoplasm. It produces ATP without any oxygen involvement. So this doesn't require any oxygen consumption. And this is how we are supposed to have used energy in early life, before you had the photosynthesis and lots of oxygen in the atmosphere and all that good stuff. In your glucose splitting glycolysis simple, solution, you are taking in some ATP. You still need energy input, right? Obviously, because it's a reaction, you're going to break things down. So you have to input some energy that comes in the form of 2 ATP molecules. Those 2 ATP molecules are utilized to break down these 6 carbon sugar into 2 molecules of pyruvate. As a result, you end up with 2 ATPs and 2 NADH molecules, which is, again, remember, another way that you can have activated carriers of energy. And that's the outcome from that one glucose split. So even though you put in 2 ATP, you were still able to get 2 net ATP out as well, okay? Along with the 2 pyruvate molecules, but there's still a lot of energy still stored in these molecules, which you can get out if you are able to use the other cycles. So where does this happen? Just the glycolysis step, it is, like I said, it's going to have your 6 carbon sugar cleaved into 2 pyruvate 3 carbon molecules. It requires 10 steps. This is already a 10-step reaction. And this initially requires the energy input from 2 ATPs. And then in that first, at the end of those first 3 steps, you have those 2 ATPs add their phosphate groups to the 6 carbon sugar, making it into a fructose 1,6-biphosphate instead of the glucose that it was. This makes it activated and ready to be broken down, right? These phosphate groups are what's going to allow it to be broken down. So each one is then going to, in step 4 through 4 and 5, are going to get broken down into those 2 3-carbon chains. These are, again, activated with the phosphate groups attached, and these are called glyceraldehyde 3-phosphate groups, phosphates, which through a series of metabolic reactions are going to release 4 total ATP and 2 NADH. So each one of your 3-carbon sugar, as it loses that phosphate that got attached to it by ATP, is going to produce 2 ATPs and 1 NADH. That's the energy generation step. So the first 5 steps are getting it activated to a form that it can produce energy. And the second half, the next 5 steps, are the energy generation component, where you utilize those activated forms to create energy, to get some energy back. Now, we had already used 2 ATPs to start the process. So your net molecule gain from this would be the 2 ATPs and the 2 NADH for each of the glucose molecules. Okay. Any questions about this portion or glycolysis?
I'm good.
Okay. So again, remember that these type of reactions typically are going to be coupled reactions because at least some of the steps in here are not energy favorable, right? They are not going to be happening just on its own. So an example of this is step 6 to 7, where, so, I can show you that up front first, where you have these glyceraldehyde 3-phosphate groups, and then they are going to be releasing an ATP molecule as a result, and you're going to be getting 3 phosphoglycerates at the end, 2, 3 phosphoglycerates at the end as a result. Well, this one, the first step 6, actually has this high energy phosphate bond that is going to be utilized for this particular process to occur. This phosphate is going to be taken away with the oxidation of the aldehyde to the carboxylic acid. That is going to lead to generation of this NADH, and that's going to be part of what is going to fuel the energy for step 7, so that you can have that formation of ATP by taking that charged 1,3 bisphosphoglycerate into ATP, leaving that uncharged molecule at the end. So the total energy shift of step 6 and 7 together is favorable because it is negative 12.5. But if we were only looking at this part, it would have been a negative step. Or it would have been a positive change, which wouldn't have made it happen. So anytime we are utilizing energy, or we are trying to move a reaction forward, we are trying to use some kind of activated carrier to create that negative delta G and to provide some form of free energy to make that process happen. The biggest way that we do that is by hydrolysis of ATP. So this is just showing you how much energy is released for various types of molecules that are present in glycolysis. So if you have a phosphate bond in that glucose 6-phosphate, right, initial phosphate, that's negative 17.5 kilodaltons or kilojoules, while a simple ATP molecule, when it is broken down, is negative 30.6. That's a lot more energy that is getting released, right, kilojoules per mole. And then when you look at those activated molecules that it created by using the phosphates from these ATP, you see that at each step, you're getting higher and higher delta G, because those are more and more unstable and very, very favorable reactions. So this ATP, by donating its phosphate groups to these other intermediate molecules, becomes a lot more efficient and is able to do a lot more work and get a lot more higher energy molecule output as a result. Now, sometimes, obviously, you don't have oxygen available or you don't have enough oxygen available in the system, in which case you still need the energy. The organism still got to live. And what they do is they switch to another component kind of that will not require oxygen while still getting some energy out of the molecules. And so that one is your fermentation. In fermentation, you still have some ATP getting produced, even though there's no oxygen around. And that is an anaerobic process, but it is not anaerobic respiration, because in respiration, you have to have the electron transport chain involved, making all those electron transfers and getting the humongous amount of energy. In fermentation, you're just going to get small amounts of ATPs released from your compounds. And basically, you're going to be focusing on your glycolysis and that initial part, and you're going to forego all that major energy exchange that happens later on in the process. So one of the ways that you use fermentation is to generate NAD plus by fermentation. So here is an example where you have your glycolysis, you got your two pyruvates, and now you do not have any oxygen around. So these pyruvates are just going to reproduce those NAD+. Those are the ones that are going to keep getting regenerated from lactic acid or lactate using the hydrogen ions from there. And that kind of oil rig reaction will produce some lactate and then will give you some small amount of energy in the form of NAD+ ions and ATPs. So the fermentation will lead to, at the end of the day, overproduction of this lactic acid and alcohol. So you're going to have carbon dioxide overload, and you'll have ethanol that is going to be produced in your system. That's going to have to then be taken care of and excreted out. Here is an example of how some molecules, many bacteria, for example, do use methods like that to run their energy consumption. So these are various groups of bacteria that essentially that's how they survive. They are anaerobic, and they are going to be using these alternate mechanisms of energy consumption or energy production rather, by using either the lactate or producing other intermediate compounds to get their energy from these systems. Many times these are going to end up with various alcohols as the byproduct that you can then utilize or that you can use. Okay, so now we got our two pyruvates, and we are ready to do a massive energy change or energy production. So there are many different types of organic molecules that can be used, not just the products of glycolysis or not just products of sugars, that can be used by our mitochondria for energy production. And that's done through the help of the mitochondrial matrix and the components within it. To do that, to be utilized within the mitochondria, they need to be converted to acetyl-CoA, or one of the components of that cycle. If you look at the mitochondria, you have several layers, right? You have your outer mitochondrial membrane, you have an inner mitochondrial membrane, and then you have these little intersections. These are your, this is the inner matrix of your mitochondria. And in between the outer and the inner layer is what we call the intermembrane space, that things can move from one place to another, okay? So starting off with the second step, we've got our pyruvates. In this case, we're just going to use our glucose as an example initially. And pyruvate is going to then be taken into that first layer, the outer membrane, and it is going to start to be converted. As it starts to enter the mitochondria, it's going to convert into acetyl-CoA by the mitochondria through a process of oxidative decarboxylation. In this process, the carbon is lost in the pyruvate to form an acetyl group. As it does that, it is oxidized and binds to acetyl-CoA, and with the help of exchange molecules like NADH+, that becomes reduced to NADH by using that hydrogen ion that pops off from this structure. Similarly, so that's, pyruvates are usually coming from your amino acids or from your sugars, a lot of them from your sugars. Fatty acids are also going to be oxidized to the similar structure, but through a different mechanism. So fatty acids are oxidized to acetyl-CoA similarly in mitochondrial matrix. They go a little bit deeper in there once they enter. And this is your typical triacyl glycerols. You have those three fatty chains attached to the glycerol. And in your mitochondrial matrix, what's going to happen is that these fatty acids are going to be released from that triacyl glycerol by activating the head, that acetyl-CoA head, with 2 phosphates from ATP. It's going to activate that, right? So this is the AMP plus those 2 phosphates, and you have your glycerol molecule. That activated version of the fatty acid chain is going to allow it to be separated from that glycerol molecule and enter into the fatty acid CoA cycle. This process, again, goes through several steps where it is activated and then reduced and activated again and reduced or oxidized completely. And these repeated cycles of oxidizations to get it to form the right component is going to release small packets of energy in the form of FADH2 and NADH. Those would be the components that would be coming from those for energy, not ATP. Does this make sense, guys, for this part? Like that. Hello? Okay, thank you, Rhoda. Okay, so the citric acid cycle, so you have your acetyl-CoA coming in, and then once those acetyl-CoAs are there, however they got there, they are now going to go into the second part of energy generation. That second part of energy generation comes in the form of citric acid cycle. And this generates energy in the form of NADH using these acetyl groups in and oxidizing them to carbon dioxide. So essentially, what we saw up here, sorry, why is not there? So right here, this is the cycle that we are seeing for fatty acyl-CoA. The same thing will happen with our pyruvates as they come in once they change into acetyl-CoA and get looped into the system. That's the citric acid cycle. So in our typical citric acid cycle, you have those acetyl-CoAs that have come in from the breakdown of pyruvates, and they're going to go through several rounds again of oxidization. And through those processes, each time they're going to be producing NADH and some GTP, sometimes as a minor component, and FADH2. So one of these cycles produces, and so each pyruvate is going to go through and turn into an acetyl-CoA, and then each one of them is going to do this cycle once. So each of those cycles are going to produce 3 NADH, 1 GTP, and 1 FADH2. And as a result, also produce some carbon dioxide as part of the respiration, right? So that's the waste that's coming off. So your net result is going to be all those NADH molecules, the FADH2, and the GTP that are released. So this tells you that there are other energy molecules activated carriers as well that we had already talked about a little bit. So just to remind you what those are, guanine triphosphate, GTP, right? It's essentially the same idea as those adenine triphosphates like ATP. It has 3 phosphate groups attached to your guanine and the ribose sugar. And these are going to have the same type of energy in there as we did with ATP. And the second one is your flavine adenine dinucleotide as electron carriers, where FAD is going to get activated into this FADH2 molecule that is again going to be a high energy bond as well and is able to release energy, not as much as ATP or GTP, but still a lot of energy as well. And this can then interact with ATP or other molecules, ADP and other molecules as well. So here, let's go over up till now what we did. We have our glycolysis that's going to be breaking down glucose from glucose 6-carbon sugar into your 3-carbon pyruvates, right, through a series of steps. As it's doing that, you are also going to be getting nucleotides, amino sugars, glycolipids, and glycoproteins from those byproducts that are getting produced at each step because they are components of those products. For example, glucose 6-phosphate is going to become parts of the nucleotides. Fructose 6-phosphate will be part of your glycoproteins, amino sugars, glycolipids. Then at the end of your glycolysis, you are also going to have a portion where your acetyl-CoA can be made from lipids and be introduced into the system, as well as amino acids can be produced from those different byproducts from steps 6 through 10. Once you have your pyruvates or you have your acetyl-CoAs from amino acids or lipids, they are going to go into the citric acid cycle, where they are going to be going through several levels of oxidization to get more energy packets out in the form of NADH, FADH2, and others. This process also is essential for making, it's coupled with many other metabolic processes like cholesterol and fatty acid production. When the citric acid cycle is going on, another intermediate alpha ketoglutarate is important for production of glutamine and other amino acids and purines. A third byproduct, succinyl-CoA, is part of the heme and chlorophyll. So each part of this cycle is also providing components or basic structures for as input, the substrates for those reactions as well. Okay, so now we got a lot of different substrates for a lot of different metabolic reactions happening inside our body. And we also, at the same time, are producing some minor forms of energy. But obviously, that's not going to be enough. We need a lot more. And for that lot more, you get to the inner matrix of the mitochondria for the most production of energy. That's why mitochondria is the energy production system through the electron transport chain. The electron transport chain is the main driver of majority of the synthesis of ATP that is going to happen inside our body, inside our cells, and it is part of what we call oxidative phosphorylation. Okay, so before that, most of the phosphorylations that were happening are happening through enzymatic linkages and not through oxidation, right? So, or not through oxygen. So the first part, pyruvates from glycolysis are going to be coming into our mitochondria and getting, those pyruvates are getting converted to acetyl-CoA, going into the citric acid cycle within the mitochondria, and then producing a lot of NADH from glycolysis, pyruvate from glycolysis. But then at that last step inside that inner matrix, you're going to have the oxidative phosphorylation step, which will take the oxygen as the electron carrier and utilize it in electron transport chain to produce the majority of ATP. This is a very, very controlled mechanism, and it is how our catabolic and anabolic reactions are actually organized and regulated, again, so that they are linked together in order to get the maximum energy output with the least amount of effort. And anytime that you do not need the food or the energy that is available, it is able to be stored in reservoirs so that it can be utilized later as needed. Here is an example, this is just like a little diagram showing you all the different metabolic reactions that might be happening inside the body. Glycolysis and that citric acid cycle constitute a very, very small component of that reaction, of all those metabolic reactions. These are all of those other ones, whether it's making nucleotides or proteins or amino acids or anything else and metabolizing those things. Those are all those other dots, all those other chain reactions that are also happening at the same time inside our body. So sometimes we have an issue and we do not have enough sugars. And so there is a process through which our body can also move these reactions in reverse. But remember, that's not going to be a very energy favorable component, and it's going to require a lot of energy input as a result as well. Gluconeogenesis is essentially a process by which the cells can regenerate glucose in the blood by moving these reactions in reverse component, right? So they're going in the opposite direction. So instead of breaking down the glucose, you're going to try and build it up again, going through all those steps in reverse. That will require the same amount of ATP and the GTP and everything getting put into the system in order to get that system moving in the opposite direction. When that happens, some of those steps cannot be reversed because it's just not possible. And those steps would then lead to the product being a little bit different at the end of the day than before. So here is an example that in glycolysis step 3, you take a fructose 6-phosphate group phosphate molecule and you activate it on both ends by adding another phosphate group to the other end to make a fructose 1,6-bis phosphate. If we were to go back and try to reverse it, we can bring it back through gluconeogenesis to that fructose 6-phosphate, but we are not going to be able to release an ATP or get an ATP. You're just going to get a phosphate group and water as a result, using water as a hydrolysis as the input. So some of the things that can be utilized to control this process are things like this. We use these intermediate molecules that cannot be changed or that can be controlled to regulate how much of sugars we are making versus how many sugars we are breaking down. So fructose 2,6-bis phosphate is a way, it's a modified version of that molecule that is an important regulator to see whether we need to run glucose and break it down and do glycolysis, or if we need to go through gluconeogenesis instead. And based on the levels of this particular molecule, along with glucagon and epinephrine, we can make the system go in one direction or another, or our bodies rather can make the system go in one way or another. So for this system, there are many positive regulators. So if there is not enough ATP, there's only AMP and ADP around, lots of phosphate groups around, it's going to run it into the positive direction and make more of this happen, more gluconeogenesis happen. On the other end, if there is a lot of ATP around, lots of citrate, fatty acids, plenty of other stores available, it's going to do a negative regulation and inhibit gluconeogenesis as a result. So that's what you're seeing over here, that you have the PFK and fructose 2,6-bis phosphate, and based on which positive and negative regulators are present, it's either going to activate glycolysis or activate gluconeogenesis. Now, animal cells will store glucose when it's extra in the form of long branched chains. These are called glycogens, and they can provide us with energy in times of need. These chains are linked together again through phosphorylation of these structures. So your glucose is essentially getting phosphorylated to create activated molecules, so they can be made into polymers. And they are going to be, they are not exactly perfectly branched. They are randomly branched structures. They are joined together to create long polymer-like structures that can be stored in the liver cells and then utilized as needed. Similarly, another way that we are going to store energy is in the form of fats in lipid droplets. These usually form inside the cells, and they are going to be using the endoplasmic reticulum that are coated with the cholesterol and triacylglycerols. And those little vesicles that form are going to be your lipid droplets. They are going to be filled with triacylglycerols and cholesterol, and on the outside will be associated proteins that typically hang out with those lipids. Plants will also do something similar. They are going to store both starch and fats in their chloroplasts instead of, in us, we have those liver cells and other stuff. In plants, they're going to store them in the form of starch and fats in their chloroplast. And here you can see that there's a chloroplast. You see these large kind of vacuole-like structures, and then you see these smaller oil droplets of fat. These are where it's going to store this energy that it can take when it needs. Seeds of plants will often also contain large amounts of fats and starch as storage molecules and provide that initial energy for these plants to get growing when it starts. So just to review, catabolic and anabolic reactions are usually linked, organized, and regulated. There are feedback regulation steps available within the system to make sure that we are not breaking down too much glucose or making too much glucose, and that we can have positive and negative regulation of them. And cells can store foods and energy in special reservoirs, not just in the form of chemical energy or bond energy, but also in the form of fats and lipids and starch. Questions? the electron transport chain and going into it more in detail. And then we shall be going from there. Okay. Okay, kids. So we'll talk with you later. Take care. Bye-bye.